do in fact form the micelles in solution. The work that we've started now, and be carrying on whilst I'm here, is to try and functionalize these. So as I said before, you can already track compounds that are otherwise insoluble inside them. And the idea is obviously your body is full of water, so that's an aqueous environment. So that's where something like a micelle will be useful to take in an insoluble compound, say a drug. But obviously it still delivers it everywhere as it flows around the bloodstream. So what we're trying to do is to functionalize these, so the green bits, so that they target specific cells within your body. So all the cells have receptors on them. This is how they talk with each other. And so if we can target those specifically, then we'll know exactly where the drug's going. Because obviously a problem at the moment is you throw in drug and it goes everywhere, but you have to have throw in a lot of that drug so that a small amount gets to the area that you need it to go to. So it fixes the problem, but also makes a wheel in the process. So if you could get a lot more of the drug specifically to that area, it's going to reduce the bad effects that you don't want to go along with that drug. How we're doing this is, you see this ene unit. It's a carbon-carbon double bond in the hydrophobic block. This is quite reactive, and so we're using this in our micelles to then, we've got our micelle dispersion. We'll add what we're actually going to add is peptide sequences, so short proteins, which will react with this bond and stick to the surface. So then, with our drug traps in the center, we should be able to send these to specific cells. Notably, cancer cells, they overexpress a lot of things relative to the other cells, so we should target them specifically. Anybody want to know actually about the chemistry of how we're attaching them? Or I'm not sure where we've got to.
So yes or no?
Yeah?
Will it make it more confusing?
Well, quite possibly. It's a relatively simple reaction. We'll should be able to control it in solution. So with this double bond, we've got that attached to our micelle, so these sticking out all over. And what we're going to synthesize is these short sequences of peptides, effectively a miniature protein. But we're going to make them with a specific cysteine amino acid on the end, which our peptide sequence, which ends with an SH group, the file. And with super initiation, this file, so we're going to add as an initiator to generate some radicals in solution, which are basically very, very reactive. And so they're going to strip this H off, and we'll end up with then a very reactive file, which is then going to attack here, and perhaps there. And so what we'll end up with is our micelle with our peptide sequence on the end, and H will add here. And so with these all around the surface of our micelle, we should be able to functionalize them. As I mentioned at the start, the different volume fractions of your soluble block and your insoluble block are going to affect the structures that form. So our peptide is going to be soluble so that it sticks out of the shell so that it can recognize cells when we throw it into the body. but in effect it will be adding extra soluble parts to this polymer chain. So it's another thing we're going to have to look at when we do this, is whether we're completely changing the structure and disrupting the system. Because we're obviously going to need to maintain that for it to be useful for in our initial plan. And so I mentioned here we will be generating the radicals, and so we can do this in a controlled way by, as with the polymerization, if you add a certain amount of your initiator, will generate a certain amount of these radicals, and so only a certain fraction of these are going to attach. And so we'll be able to make a series of these with different amounts of functionalized groups attached to the outside, and see the point where we start to disrupt the system and make sure we're maintaining our micelles.
Did you have a question?
Oh yeah, sorry.
So you can get the micelles to attach to any other sort of cell, like into like a body cell.
Yes, you can, but it's harder with just cells in general that aren't ill, because the proteins, basically they express proteins on the surface of the cell. There's going to be proteins sitting in the membrane, and that's what we're going to attach to. And then once this micelle is attached, the cell will recognize it and sort of engulf it and take it into itself. But in general everyday life, they're kind of expressed about the same. So it's not so easy. But with cancer cells, there is, I forget the name, but a specific protein that they overexpress, and so a higher portion of the micelles will then target the cancerous cells rather than the normal cells in solution. Yeah.
So once they attach to a cancer cell, is the goal to get the inside of the micelle to interact with the cancer cell?
So the plan would be, if once it sticks to the surface of the cell, it's going to get engulfed within the cell. And so this particular polymer won't do it, but when a cell engulfs something, as I showed before, it's like the big membrane, and when it takes things in, a bit of that membrane sort of peels off, and so the micelles it's taken in are then trapped within that in another membrane. The way it goes to release that within the cell is there's a drop in pH. So the normal cell is about pH 7 neutral, like the rest of the body, but it drops to pH 5 with salts, and water will then come in, and that will rupture, releasing, in this case, the micelles. If you were to make this portion of the polymer so that it was responsive to a change in pH, then when that was released inside the cell, say a drop in pH would add, if you had an amine group, so a nitrogen on the end, you could protonate this, it would become charged. This portion would then be soluble in water, and so the micelle would then break up in the cell. So that's another plan. And that's then how you could deliver your drug into that specific cell, which would then hopefully kill the cell or help the cell if you were going down that route instead.
Anybody else?
I think.
Cool.
That's sort of work I'm up to at the minute. If you have any questions going back through that,
Okay,
please feel free.
let's thank Thomas.